The middle is kind of darker. We had an example of stuff like that yesterday where if the stain is darker, more towards black, that means there's less signal. And so there isn't really actin inside the nucleus, but it really hangs out and helps us organize and makes the shape of the cell. So it organizes the plasma membrane. Actin can interact with myosin, which is, of course, we probably all know this, involved in muscle contraction, where the actin and the myosin crawl along each other, or rather the myosin crawls on the actin. The myosin is a motor protein, so it can use ATP and move, and it moves on a substrate, and that substrate is actin. The microtubules, those are the fattest tubules in our cell. They really start off from the nucleus. That's where they start their organization. And so you can see there's this really high density of microtubules around the nucleus. What they do there, they help to separate the chromosomes during mitosis. So they form those protein structures that really physically help pull apart the chromosomes so that the daughter cells each get a right complement of chromosomes. They can also interact with motor proteins, different ones. The kinesins and the dyneins, those are going to be transporting vesicles through your cell. So you'll have a little motor protein that walks on the freeway of microtubules, and really taking a big vesicle of stuff to different places. So remember the vesicles budding off from the Golgi or the ER, those are taken on microtubule freeways by those motor proteins. And we'll see a really cool movie of how that works. So the intermediate filaments, they really are structural support proteins. So yesterday we talked about the nucleus and how there's this laminin on the inside, right? The inside coating of the nucleus. Those are intermediate filaments. And then also keratins, the stuff that makes your hair and your nails. Those are intermediate filaments. And that's one more comment. These might seem more boring, but they're really super important, and we'll see a disease associated with having improper intermediate filaments. So here's just some pretty pictures that really show you the difference in the localization of these proteins. This is the type of stuff that our lab loves doing. Do you guys know how such pictures are made? How do we get the colors on tubulin and on the microfilaments or on DNA? No? So anyone in the lab that does this? Jody's in our lab. Sorry, Jody. So there's several ways that we can get these colors in the cells. Usually we first have to fix the cells and kill them, but then we can use antibodies to detect the different proteins. Do you guys know what antibodies are? Right? These are molecules that your body makes against disease-causing agents, and it recognizes a small piece of protein usually. So you can make antibodies against microtubules, against actins, and then you put a fluorescent label on those antibodies. If you put the antibodies on your cell, the fluorescent label will stick there where the protein is that the antibody recognizes. Right? And so we can see here microtubules sitting around the nucleus and stretching out all the way to the periphery of this cell. Whereas the microfilaments made of actin are really sitting in the periphery and helping shape the way this looks, but they're not really used here in closer to the DNA. For the DNA, we often have these dyes that just sit in the groove of the DNA and that will fluoresce. And we use those if we run DNA gels, but we can also use them here. Another way that we can do it is to make genetic changes where we encode a fluorescent protein that attaches to the microtubule. So you'll have the microtubule that can form the long polymers, but there will also be fluorescent molecules sticking out to the side that are not in the way of making the microtubule. Those we can get from jellyfish, right? Green fluorescent protein was isolated from jellyfish, and now we have red, we have cherries, we have tomatoes, we call them all sorts of fruity names for the different colors. And these are the three basic colors that we use a lot: blue, green, and red. You can also use a far red that we can't see, but our microscopes can. And you can show that in white or different colors. But it really gives insight into how things are organized. Here's another picture, really making it even more explicit that there's a very different spatial distribution of the different microfilaments. All right, so now, let's see if this works. Yes. Alright, that's the presentation. So, we talked about microtubules and microfilaments. Let's see if we remembered all my yada yada. Microtubules and microfilaments are similar in what aspect? A few people are thinking of the intermediate filaments. And they're still thinking about it. That's really interesting. The microtubules and microfilaments. That was better. Are both built out of monomers, whereas those intermediate filaments are not. And the intermediate filaments really were giving structural support for the nucleus or creating barriers and keratins in our nails and hair. Okay, get that out of the way. And so the cytoskeleton is used, we already alluded to that a little bit, for transporting things, to make the shape of the cell. And you can see here in this picture of a couple of cells that it's really very neatly organized. We have a couple of cells. Here is a half cell, here's a whole one, and here's the next half one. And you can see that the apical surface of the cell and the basal surface are really different. The basal side of the cell sitting on this basal lamina, so it gets some cues from the proteins that it is attaching to. This end of the cell might be sticking out into the gut lumen, for example, where we have those microvilli. They really help increase the surface area of the cell so that it can absorb a lot of nutrients through this larger surface area. And those microvilli are supported by actin molecules, actin filaments. Whereas if you look here we find those microtubules as well. And here's something that we didn't really talk about yet, that these minuses and pluses. Those hold for both the microfilaments and the microtubules. They have a minus end and a plus end where you could add protein to new monomers to both ends for actin and mostly to the plus end for the microtubules. But it's really concentration dependent what happens to those ends. But it prefers to grow from the plus end. So the new monomers are preferably added to the plus end of those growing tubules or filaments. So they're used as protein highways. This is summarizing a couple more components of what we want you guys to realize for the skeleton. We already saw, right, that this must be more dynamic than the skeleton that we have in our cells. That also remodels, but it takes weeks to months. This remodels over the course of seconds to minutes or hours, really much, much faster. And it has to do with the way that these filaments are built up. So we have the actin cytoskeleton, has a plus and a minus end to each filament, not necessarily to the skeleton as a whole, but each filament has a plus and a minus end. A new actin monomers are added at the plus end as well as the minus end, but the minus end, it happens slower and it requires a higher concentration of actin to really work well. We can inhibit or disrupt the actin cytoskeleton with this drug, cytoskeleton B, and so that would give us a tool that we can use to study what the cytoskeleton does. Actin interacts with this plus end directed motor protein. That means that if myosin connects to actin, it tends to crawl towards the plus end of the actin. This is also the end where the actin is growing, right? So if the myosin crawls at a good enough pace, it will continue to crawl because the actin continues to grow. If it crawls too fast, it might fall off because the actin isn't growing as fast. Microtubule skeleton similarly has a plus end and a minus end. There too, preferably added new dimers to the plus end, and this cytoskeleton protein is inhibited by the drug colchicine. So that would disrupt the formation of those polymers. You would end up with more monomers. This one has two different kinds of motor proteins associated with it. One that goes to the plus end and one that goes to the minus end. And you can imagine that if you transport things to the cell, you sometimes want to get them all the way out from the nucleus to the periphery. And sometimes you have to take stuff all the way from the outside to the inside. So it all depends on what motor protein you hook up, which way it's going to go. And so we have these neurons in our body that go from the end of your spinal cord all the way down to your feet. Might be, I don't know, what is this, a meter long? So we have those meter-long microtubules sitting there that are going to be transporting things all the way out to the end, the neurotransmitters that are being made and all that good stuff. So the cytoskeleton is very dynamic, we talked about this, and it's involved in different processes that are very important for the cell. So the making of the shape of the cell, the way that the cell is either stretched out a lot or balled up. We see if cells divide that they usually retract all their protrusions, become more round, then they divide, and then they settle down and spread out again. Some cells move. We have migratory cells in our body. There's macrophages that are crawling around in your tissues trying to find stuff that needs to be destroyed or eaten up, or phagocytosed. They need to move, and they can only move because they can change their shape using the change in the cytoskeleton. So that's how they move from one place to another. We need to transport things in the cell, and cell division also relies on the cytoskeleton because we need to separate first the DNA, and then we also need to pinch off the cytoplasm, right? So for that we would need actin. So there's many different diseases associated with defects in the cytoskeleton. Here's just a couple that I didn't want you to memorize at all. And they involve proteins that we haven't really talked about, but we did talk about this disease, multiple sclerosis, in the context of neuronal transport of information. And then we said that it was mostly a disease of the loss of that myelin wrapper, right? The conductance was slower. But the loss of the wrapper also means that there's more of an opportunity for ions to come in and out of the cell. And so an excess of calcium and a dysfunction in axonal transport contribute to multiple sclerosis. So now we'll talk about actin in a lot more detail, and we'll probably spend a good chunk of the next lecture just talking about actin. So here we have another picture of actin as it occurs in the cell. This one here is made by an electron microscope, black and white, and you can tell much more detail. Right here we're at 100 nanometer scale rather than where we were before in the micrometer scale. So this is now a single actin filament, which is schematically drawn here. We already talked about that it localizes somewhat to the periphery of the cell, that it's used to make those microvilli stand up. We also see if you stretch cells repeatedly, they don't really like that. So they try to fortify themselves, and they make those stress fibers out of actin. But if you just have a loose bundle fiber in your cell that doesn't connect anywhere, it's not going to help you prevent being stretched. So that's why those green proteins are used to attach the microfilaments to a protein that sits in the cell membrane, and that maybe attaches to something else, a substrate or another cell. So now you really have the structural component that can help maintain the shape of this cell. And then the last picture is an electron micrograph of a slice through muscle, where it's a really ordered structure of a lot of actin and myosin filaments that are going to be sliding alongside each other, making the tissue shorter, and that's how muscle contraction works. So actin is important stuff. I guess that's the take-home message. A little bit more detail. We talked about G-actin and F-actin. G-actin is the globular form that isn't incorporated, so that's a monomer. And then the F, or filamentous actin, is when it is built up into this microfilament. A lot of the protein that you have in your cell is actin, and we actually use it kind of as a housekeeping gene, like an internal standard that doesn't really change with whatever the cell does. So if we look at gene expression, we often compare it to actin as something that doesn't change when we treat the cells. So that forms our internal control. It's an ancient protein. Bacteria have it too. We have multiplied it, so we have a couple of different isoforms that probably specialize a little bit. That's usually what happens when we make multiple copies of something. Actin, even though it's globular, so kind of rounded in shape, we do see that it has what we call a pointed end and a barred end. And this pointed end is where actin binds to ATP. And the fact that actin binds ATP is really important for how these filaments are growing. And so this here is the key feature of, I think, what we talk about today. Actin binds ATP and can hydrolyze it, so split it into ADP and free phosphate. It makes a big difference for how actin acts. So actin bound to ATP really likes binding other actins. So you can imagine that actin ATP really helps form the microfilaments. Whereas if actin is bound to ADP, it doesn't really like binding to other actins. So that would mean that there's a bigger chance of the filament breaking apart again if it's bound to ADP. And so This process, going from ATP to ADP, actually happens pretty shortly after the actin is incorporated into the filament. So you have actin ATP, it likes to bind, it binds, and then pretty shortly after it hydrolyzes ADP, and so now it no longer likes to bind, right? So you get this dynamic changeover of how stable the filament would be. Actin always organizes itself in the same way, so the pointed end sits here towards the minus end of the molecule, and the barbed end points towards the plus end. What do you guys, I'll tell you, the difference between the minus end and the plus end. So the minus end, we said, grows more slowly, right? Actin monomers are preferably added to the plus end, and that has to do with the way that this face of the actin molecule is happier to accept the addition of new molecules, whereas this side, it requires more of a change in shape, so it's less efficient. What that means, we'll talk about this.